Friends, welcome once again in this new series in which we will learn some fundamentals and techniques in molecular biology through problem-based learning. I have included a few interesting problems and wish that you solve them simultaneously as you learn. Let's start with the problem based on DNA structure. Confused? No problem. Let's first learn the DNA structure, then attempt the problems. DNA structure is the center point of learning molecular biology. Therefore, we must know this structure before we proceed to more complicated concepts. We have always seen DNA as a double-stranded helical macromolecule. Let's learn how it is formed. DNA or deoxyribonucleic acid is a polynucleotide made up of multiple nucleotide molecules which join each other by phosphodiester bonds. Free nucleotides are also known as DNTPs or deoxyribose nucleotide triphosphate. Each nucleotide has three components. First component is a 5-carbon sugar which is ribose in RNA and deoxyribose in DNA. We can see the difference in this figure as the second carbon position or 2' of sugar is attached to an OH group in ribose sugar, whereas it's a hydrogen group only in deoxyribose sugar. Also note that the OH group at 3' position is very crucial to attach one nucleotide to another. The second component is 4 types of nitrogenous bases. 2 of them are purines, which are adenine and guanine. They belong to double ring structures, whereas cytosine and thymine are single ring structures termed as pyrimidines. These bases attach to deoxyribose sugar at first carbon position or 1' via a glycosidic linkage. Adenine and thymine are complementary and cytosine and guanine are complementary. Therefore, number of A bases will be same as number of T bases and number of C bases will be same as number of G bases. The phosphate molecules are the third component. A free DNTP molecule has 3 phosphates attached to the 5th carbon or 5' position of the deoxyribose sugar via phosphodiester bond. With all the 3 components, this molecule is termed as a nucleotide or DNTP, where if only sugar and nitrogenous bases are present, then it will be known as a nucleoside. This is the structure of a nucleotide or DNTP, where N denotes any of the 4 nitrogenous bases. One nucleotide is joined to the next one by DNA polymerase enzyme. The 3' OH group of the first nucleotide and the phosphate group of the on 5th carbon of the next nucleotide involve in creating a covalent phosphodiester bond by eliminating a water molecule. This is how a polynucleotide DNA molecule begins with 5' phosphate of the first nucleotide and ends at 3' OH group of the last nucleotide. And the direction of DNA sequence is also shown from 5' to 3'. This joining creates a polynucleotide sequence like a long chain, which will be a single strand of DNA. Since all the sugar and phosphate molecules are the same, but the nitrogenous bases may change, therefore a DNA sequence is actually a sequence of these nitrogenous bases. DNA molecules are double-stranded. So, 2 single-stranded DNA molecules have to join. This is only possible if 2 single-stranded DNA strands are complementary to each other. Then the 2 strands join up by hydrogen bonds. 2 hydrogen bonds are formed between an AT pair and 3 hydrogen bonds are formed between a CG pair. The double-stranded DNA now folds in a 3-dimensional structure like shown here as Watson and Crick's right-handed bDNA model. DNA is naturally a right-handed helix. Let's look at some measurements. The diameter of DNA molecule is 20 angstrom. Each base pair makes a 36-degree angle with the next. Hence, to make a 360-degree turn, we need 10 base pairs to be present in one helix turn of DNA. Each base pair has a size of 3.4 angstrom approximately. So for one turn of DNA or for around 10 base pairs, the length of DNA will be approximately 34 angstroms or 3.4 nanometer. After understanding some basic concepts about DNA structure and size, let's try solving these problems. These problems are based on your knowledge of DNA size, number of bonds, complementarity, etc. Either you write your answers in the comment section or check the answers and explanations in the beginning of my next video, which will be about Sanger's DNA sequencing using PBL. Hopefully you liked today's tutorial and learned something new. If you like the PBL based approach, please like and subscribe to my channel so that I keep making new videos for you all. In my next video, you will be finding the answers and explanation of these problems, as well as you will be learning Sanger's DNA sequencing method through PBL and data analysis. So don't forget to watch. Till then, thank you and bye-bye.